The Dr. Xiao integrates paleobiological, sedimentological, and geochemical data to shed light on important evolutionary events, their environmental context, and their geobiological consequences. Originally from China, Shuhai received his undergraduate and MSc at the Peking University in Beijing. He went on for his master's and PhD at Harvard with Andy Knoll, where he extensively studied three-dimensional, exceptionally preserved microfossils from the Doushantuo Formation in South China. These include algae and some of our earliest evidence for metazoan embryos. After receiving his PhD, Shuhai held an assistant professorship at Tulane University before moving to his current position at Virginia Tech, where he remains today as professor of geobiology. Throughout his career, Shuhai has worked on Cambrian, Neoproterozoic, and older Precambrian rock, but he's continued to focus on developing our knowledge of Ediacaran fossils, geological, and chemostratigraphic history with a focus on understanding the changes for life and its environment across the Ediacaran-Cambrian boundary. He's worked on Ediacaran trace fossils, algae, acritarchs, and more, and has contributed to our knowledge of Ediacaran redox conditions, the Shurim excursion, and the glacier's global glaciation. But his accolades are far more extensive than for me to even begin, so I'm just going to skip to the good part. So without any more, thanks Shuhai again for joining us today, and the stage is all yours.
All right, thank you very much. Thanks, Alex, for putting together this wonderful seminar series. I would like to mention that Alex and his advisor, Andre Beckel, are both alumni of Virginia Tech. So I'm very proud of you guys. So today I'm going to talk about terrestrial life in the aftermath of a snowball Earth. And hopefully by the end of my talk, I will be able to get rid of the question mark at the end of the title of my talk. I'm speaking on behalf of my co-authors. Some of them are shown here, but a full list of them is in this listed here in this paper that was published a little while ago, early this year. But I'm going to talk more than just this paper. There's a little more data and a little more information that was not included in the paper. So the motivation of this study is to explore the terrestrial life in the Precambrian. Terrestrial life, it's important in modern Earth. Terrestrial biosphere accounts for more than half of the bioproductivity and accounts for a great majority of standing biomass on modern Earth. And as shown in this cartoon, terrestrial life, including land plants and microbes, play a very important role in soil development, nutrient cycling, silicate weathering, geochemical cycles. This cartoon illustrates how microbes and tree roots can release important nutrients such as phosphorus and nitrogen and participate in also very importantly in carbon cycle. When we talk about terrestrial life, we often think about the rhinichirds, Devonian, but terrestrial life is more than plants, and is older than commonly thought. So this is a phylogenetic tree of bacteria published last week in Science. What it shows is the deep divergence between two major groups of bacteria. And one of them is called Terrabacteria, which is a deep bacteria branch, and many of them have terrestrial adaptations. And a molecular clock analysis indicate that terrestrial Terrabacteria diverged at least 2.8 billion years ago, indicating that perhaps they either evolved or many of them had a terrestrial lifestyle back in the Archean time. Among all the bacteria, perhaps the most important bacteria group to geologists is cyanobacteria. Obviously, this is because cyanobacteria makes oxygen, and it played a very important role in the redox condition of the surface Earth. through a technique called ancestral character reconstruction. Molecular biologists are able to reconstruct based on the character distribution and the topology of the phylogenetic tree, reconstruct the character of ancestors. And in this case, they are trying to reconstruct the habitat of the last common ancestor of cyanobacteria, and it turns out to be non-marine or freshwater. Not only cyanobacteria, perhaps started from a terrestrial environment, but also the plastid, the eukaryotes that first evolved the plastids. So they evolved the plastid from cyanobacteria, including red algae and the green algae. And this study shows that the last common ancestor of red and the greens were also freshwater organism. One may criticize that there's some sampling bias here because it's easier to sample terrestrial cyanobacteria and the terrestrial algae than marine ones. So perhaps I don't have a way to tell, but it's possible that there's some sampling bias here. Fungi, another major group of organism, eukaryotic organism, it's almost entirely on land, and many of them are fully terrestrial, including things like this major group here, your yeast, truffles, and morels. This group is your mushroom. So there's some technical name, Basidiomycota and Ascomycota here, and this two group, which do not form a monophyletic group, that's why it is in quotation mark here. Forms the zygomycetes. All the three major groups are fully terrestrial. They lost their flagellum, so they don't have their reproductive cells do not move in aquatic environment. Instead, they reproduce by spores. Even the low fungi, many of them are aquatic, but they are freshwater. So the molecular clock study indicate that the fully terrestrial fungi started at least in the Tonian or perhaps or in the Cryogenian. But the fossil record of terrestrial life is really far in between, and many of them are controversial. This is a review paper by Wellman and Strother, where they list the examples of Precambrian terrestrial fossil record. You can count them. They're in green labels. You can count them on your fingers. There are not many in the Precambrian. So we are facing another type of bias, different from the molecular biologists who might have sampling bias toward terrestrial organisms. We as paleontologists, particularly Precambrian paleontologists, have a sampling bias toward marine record because there was simply very little terrestrial record in the Precambrian. So here are some examples, a couple of examples from the literature about Precambrian terrestrial fossil record. On the left, this is some structure published by Greg Retallack. I saw that Greg is in the audience. From the 2.2 billion year old Paleosaur. So he interpreted this as some soil organisms in the Paleoproterozoic. On the right is a study published by Botryoids and Paul Knopf in 1994. Some filamentous microfossils from the Mesoproterozoic mescal and the Tonian back spring. And I think I'm convinced that they are microfossils. They are from paleokarst deposits, so they're interpreted as terrestrial organisms. But today I'm going to focus on the Ediacaran. And the Ediacaran, it's even more important to understand terrestrial life. In addition to all the reasons that I discussed earlier, there's this additional problem of CO2 drawdown, and thanks to Paul Hoffman, who provided the reference to me. So this diagram from 2012 shows many models that estimate a time scale for CO2 drawdown after the snowball Earth. And you can see there's an uncertainty of 4 or 5 orders of magnitude. And very few of them consider the role of terrestrial life in silicate weathering, despite they were very important, at least in modern environments. So I think it's important to understand terrestrial life in the early Ediacaran, not only because to tell us how microbial life in the terrestrial environment reacted or responded to the snowball Earth, but also it can help us to constrain or better parameterize the geochemical models. Okay, with that, I want to take you to South China. So this is a map of South China, and many of the sections of the Doushantuo Formation, which is better known for the embryo-like fossils, algal fossils, and aquitox, and also shuram and things like that in the Doushantuo. Here, this is a number of sections, stratigraphic column of the Doushantuo Formation from the inner shelf, the shelf lagoons. So this is inner shelf, the shelf lagoon sections, and the outer shelf and the upper slope facies. But my focus today is at the base of the Doushantuo Formation that is highlighted brown here. The capped dolostone that was deposited immediately after the terminal cryogenian diamictite of the Nantuo Formation. The capped dolostone is no more than 5 meters in most of the sections. And it's constrained by 2 radiometric dates that are basically indistinguishable from each other within the analytical uncertainty. So that constrained the capped dolostone to be 635 many years old and lasted less than many years. 2 of the important features of the capped dolostone that I want to focus on today are the you see that on the top of the capped dolostone, there are these sawtooth structures here that illustrate the castification surface on the top of the Doushantuo capped dolostone. And another feature that I'm going to discuss extensively is this lensoidal structure within the capped dolostone that represent the sheet cracks or sheet cavities that I'm going to discuss. So this is a typical outcrop of the Doushantuo capped dolostone in the Yangtze Gorge area, maybe 3 meters in thickness. This is exactly where this age 635 age came from. This age did not came from this locality, but is correlated to here. So together they constrain the age of the capped dolostone. In almost all the sections, you see evidence of castification or caustic dissolution at the top of the capped dolostone. So this is manifested as this uneven top surface and is overlapped by the overlying strata. In this case, is thin bedded dolostone on the left, and on the right, this is overlain by phosphorite. And again, the topographic relief up to 1 meter or more. Sheet cracks or sheet cavities, they're called so because they typically follow the bedding. So here's some of the sheet cavity that is filled with silica or quartz. So they're more resistant to chemical weathering. And this is, again, the sheet cavity or sheet crack following the bedding in a TP-like structure. Again, it's silicified. So the formation of the sheet cracks are thought to be, they were physically initiated because either the increase of the pore-fluid pressure or the decrease in the hydrostatic pressure. But I think on top of that, chemical dissolution, at least in South China, must have played a role to augment the sheet crack or sheet cavity. So for example, here in cases like this, you can sort of remove the sheet crack and put the blocks of the capped dolostone together, so you can recover or reconstruct the pre-cracking morphology. But in cases like this, you have a cavity here that probably requires dissolution, chemical dissolution. And our hypothesis is that that is probably related to the karstification event, and it's related to the post-glacial rebound based on the time scale of the chemical dissolution. So it's not, it probably lasted 10 to 100,000 years or that order. So because chemical dissolution also involved in the formation of this structure, so we're going to call them sheet cavities. So this is a close-up a picture of one of these sheet cavities. As you can see, so the boundary, so this is capped dolostone here. This is the boundary between the capped dolostone and the sheet cavity. Here's the upper boundary. The sheet cavities are typically filled with several generations of cements. Typically, almost always you have a first generation of what is known as isopachous dolomite. So this is a dolomite. The chemistry of the dolomite is somewhat similar to the capped dolostone. And then in this case, there's another generation of what is known as radiochalcedony, as silica, and then another generation of coarse quartz. So this quartz crystal pretty big in this case, and then it's symmetrical on the other side of the sheet cavity. You have the radiochalcedony and the isopachous dolostone. In many other cases, you also have additional generations that are listed here. So this is a full list of the different types of cements: fibrous calcite. Sometimes you have blocky calcite that are not present in this particular example. In thin section, so you can take a look at what these cements look like in thin section. This is the isopachous dolostone. So this would be the capped dolostone. This is the isopachous dolomite. This is several generations of siliceous phase, including the RC, radiochalcedony. The point I want to make here is something that will come back later, and that is in the siliceous phase, including both the RC and the CQ, so the coarse quartz. Sometimes they preserve microfossils. If you have good eyes, you can see some filaments in this box here, but we can come back and I'll show you much better pictures later. But just keep in mind that the fossils are preserved in the siliceous phase. Now, I mentioned the fibrous calcite, but I didn't show you any examples of fibrous calcite. And that's because the fibrous calcite FC is typically replaced by the siliceous phase. So this is a CL, a cathodoluminescence image of a sheet crack. So you have the capped dolostone here, the ID, and the radiochalcedony. Radiochalcedony is black, and then the ID. And then when you in the cap again. So when you look at the boundary between the cap and the dolostone, so this is the BSE image of one of these boundaries between the cap and the ID, the isopachous dolostone. Sometimes you find a thin layer of stuff that is enriched in clays, and we think that is because they represent the residual from the dissolution of the capped dolostone that form the sheet cavity. So this is dissolution residual, and then on top of that, you precipitate, in terms of cement stratigraphy, you precipitate the ID, which is this, this bright face on the CL. And then you precipitate the FC, and then the FC is partially replaced by the siliceous phase. Now, I say that because there are cases you can clearly make out the perigenesis sequence. So this is one of the examples. You have the cap dolostone here, the microspar of dolomite, and then you have the isopachus dolostone with very beautifully preserved crystal termination. No evidence of dissolution here. This is a point I'm going to come back later. And then you have this is a fibrous calcite, and the fibrous calcite, and some of the layers are replaced by the radial chalcedony. So this is siliceous phase. Here's same sort of phenomena. You have this is a transmitted light. This is under cross polar. So this highly birefringency color, that's your calcite, the fibrous calcite, and it's partly replaced by the chalcedony. And sometimes the lamination is partially preserved, for example, here. Not only the fibrous calcite, and here again, fibrous calcite is partially replaced by the radial chalcedony. Not only the fibrous calcite, but also the isopachus dolomite can be sometimes replaced by the siliceous phase. In this case, the entire layer of ID was silicified. In this case, you can see at the base of the ID, there's some silica that we think is secondary replacement. And finally, so you precipitate this siliceous phase. There's a study of fluid inclusion in the quartz in the siliceous phase, indicating that it is hydrothermal. So the homogenization temperature is 200 degrees plus minus. And finally, the residual pore space is filled with this blocky calcite here. So blocky calcite, and you can see this higher birefringency in the center of the sheet cavity. That's the blocky calcite. You don't always have the blocky calcite, but when you have them, and if you analyze the carbon isotope, it typically has minus 40, minus 50 per mil of isotopic signature. So here's our reconstruction of the perigenesis. So formation of the sheet crack, and then you precipitate the ID, and then the FC, this laminated fibrous calcite. And then there's a silicification and a precipitation of this quartz grain here. They're related to your hydrothermal activity. And finally, the blocky calcite in the middle. Okay, so another important, so that's the first part of the talk, the cements and the perigenesis. The second point I want to make in the next few minutes is that many of the fibrous calcite and the radial chalcedony, they make speleothems. So this is a hand sample, and I need to explain how this hand sample has 3 cuts. So it's like a step. So this is a rhizome of the step, the tread, or the rhizome and the tread again. Or you can think this as a chair. So this is the seat of the chair and the back support of the chair. So we have 3 cuts, 2 vertical cuts, and this is stratigraphically oriented, and a 1 horizontal cut, right? So you can see there's some darker color structure here. You can't see what they are at this scale, but I'm going to illustrate what they are in a magnification. Some horizontal cross section and a vertical cross section that hang down from the roof of the ceiling of the sheet cavity. So when you take a closer look at this columnar structure that rises from the floor of the sheet cavity, you notice that there's all this radial chalcedony structure, right? So it rising from the floor, columnar structure, and it's surrounded by these quartz grains. You can also take a look at the transverse cross section at this horizontal cut, and you see this concentric layers. Some of them has a little central tube in the middle. For example, here, some of them sort of aggregate together, and those are the epifluorescence image showing the laminae, gross laminae. So we interpret them as a microscopic stalactite that hang down from the roof, or stalagmite that rising up from the floor. And some of this stalactite has a central tube that is in speleothemology is called soda straw. Okay. Here again, there are more examples of stalactite hanging down from the roof. You can't really see much of this because this magnification is not good enough. But you have to take my words. This is a stalactite touching a stalagmite, and it becomes one column. And in a term of speleothemology, this would be called a stalagnate. So this is a close-up thin section of some of the stalactite. Again, the central tube or the soda straw in a vertical or oblique. This, I believe, is an oblique section and a more or less transverse section, and epifluorescence image of the same thing. They're all silicified. Here more example of this stalactite, but I put it against some modern analog of stalactite. So this is a modern soda straws. So this very thin cylindrical structure. They're the feeding channels of the dripping water. So when you have a drop of water staying at the tip of the soda straw, as it degas and evaporate, it forms a rain of precipitate. So that straw grow over time, and eventually it would be clogged. But there's also overgrowth from outside. So for example, here you have this structure that is growing around the soda straw. And some of the stalactite, you can see the soda straw in the middle and it's still dripping water, but you also have seeping water from the surface and precipitating more laminae from the outside of the soda straw. I think this is what happened here. Some of them conical, for example, this one, the modern ones are also conical. There's a third type of a speleothem that are more irregular in shape. Some of them are oblique, some of them are horizontal, some of them are twisted, some of them branched. And you can see this case, this is an oblique speleothem. So in speleothemology, it's known as a halictite. And I'll show you a modern example later. This is thought to be formed by, mostly influenced by capillary flow and the dynamic airflow in the cavity. So this is an epifluorescence image of some of the halictite, and this is a SEM image of this area labeled here, showing the presence of a central channel and the growth laminae around the halictite. This is a modern analog of halictite, and you can see that they're not always vertical. Some of them are vertical, some of them are horizontal, some of them branch, as do the Doushantuo example. Some of them are oblique. They're mostly driven by seeping water or capillary water, and their shape is modified or influenced by dynamic airflow in the cavity. So here's our interpretation. I'll propose the perigenesis. We think that they're related to the post-glacial rebound on local sea level drop, resulting in subarea exposure and castification, so epikarst, and the formation of sheet cavity. We think that the sheet cavity is formed in the fluidic zone. So the fluidic zone is below the water table, and is where the groundwater derived from meteoric water mixed with groundwater derived from seawater. Hence, that explains some of the marine signature of the ID. So the ID of the first generation of cement precipitate in the sheet cavity. Perhaps the magnesium for ID precipitation was supplied by the dissolution of the capped dolostone. So it inherited some of the geochemical signature from the capped dolostone and also from the seawater, marine water infiltration. And then sea level continues to drop and this cavity is raised above the water table into the vadose zone. So the chemistry changed, the precipitation, different kinds of precipitation. So starting with the fibrous calcite and a speleothem formation in the vadose zone. And we think it's during this time that the microbial colonization happened in the cavity. And then everything is silicified, plus there's additional silica precipitation in the cavity. Those are the purple color here. And then finally, there's a precipitation of the blocky calcite that we don't know when, but it has this very distinct, dramatically negative carbon isotope signature. So I kept telling you that all this has to do with the post-glacial rebound, but I didn't really offer you any evidence for that. So the evidence comes from this several lines of argument. If we just look at the cavity, the cavities must postdate the capped dolostone. That's the only thing you can infer, and the cements must postdate the cavity. But it doesn't tell you when, other than it's younger than 635. It could be Triassic, right, in theory, when South China is raised above sea level, or it could be a decade ago when Barack Obama was the president. So we have to constrain the age of these precipitates. The first line of evidence coming from the fluid inclusion in the quartz. I mentioned this, the homogenization temperature, 200 degree plus minus, and that's also consistent with the Raman geothermometry of carbonaceous material that gives you 200 degree plus minus. So it cannot be Triassic or later, because South China has not been buried in great depths after the Triassic time. We also directly dated the isopachus dolomite. That gives an age of 635 plus minus 17. So this is, the uncertainty is not great. This is in situ dating using laser ablation, ICP-MS, and is using the uranium lat dating technique. But it's consistent with our interpretation that this has to do with the post-glacial isostatic rebound. Another feature that I mentioned earlier, but really didn't talk about in great detail, is the perfect preservation of the crystal termination of the ID. And you can see this here. This serves as a nucleation site for the next generation of precipitates. No or very little evidence of dissolution. That means, in my mind, that means that the precipitation of FC followed very closely after the ID. Because if it's millions of years after, then you would expect to see some degree of dissolution of the crystal termination, right? And final lines of argument is that all this classification evidence stops at the top of the capped dolostone. There's no sedimentological evidence for classification in the overlying unit of the Doushantuo Formation. So we think the most parsimonious interpretation is that the isopachus dolostone and the fibrous calcite and the speleothems are all related to the post-glacial isostatic rebound, or the sub-area exposure related to that. We're not quite certain when silicification and the hydrothermal precipitation happens, and not certain when the blocky calcite happened. But this does not change the conclusion of our study, and that is the speleothem and the fossils preserved in the speleothem that is later silicified by the silica. So I mentioned the fossils a few times, but I've not really showed you any pictures of the fossils. So the fossils only come from Wun'an, and so this is one of the many localities of the Cap Dolostone that I talked about. So now let's take you to Wun'an. That's where the fossil come from. Really, we are not looking at outcrop. These are all mining pits, because here the Doushantuo Formation consists of 2 units of phosphorite that has been mined since 1950s. Gigantic phosphorite mine here. So you have the Cap Dolostone here. This is 1 unit of phosphorite that is black in color, and that's another dolostone unit. And above that, there's another unit of phosphorite that's completely gone. What you have here is just mining talus. This is one of the sections, one of the 2 sections where the fossil coming from in the Wun'an area. Again, you have the Nantor diamictite. I think you can see some of this is a cast of one of the clasts. And the Cap Dolostone and the black phosphorite that is mined in the pit. They dug tunnels into the mountain to take out the phosphorite. And the middle dolostone, another phosphorite, and the Denyin Formation above. All right. Okay, so when you make thin section of this sheet cavities, the speleothems in the sheet cavity preserve filaments like this. So you can see some of the concentric deposit here. But I'm going to focus on these filaments here. I'm going to show you some more close-up photographs of these microbes. We interpret this as fungal fossils. And just to make the point that it's not crazy to have fungal organisms in a cave. On the right, this is a modern analog of fungal hyphae within and on top of modern speleothems. So very thin filaments, 1 or 2 micrometers in diameter. Some of them are coated with precipitate. Some of them are just carbon. Some of them branch, just like the filaments we have from the Doushantuo. So take a closer look. The thicker filaments and the thinner filaments, the diameter range from a couple of micrometers to up to 10 micrometers. There are also these spherical structures that are associated with the filaments. So a few of them are highlighted here by the double arrows here. This is just about 20 micron in diameter. Most of the filaments, they branch in various different ways. Some of them branch like a Y, some have T-shaped branch, J-shaped branch, A-shaped, H-shaped branch, and a ladder-shaped branch. So the branch, some example here. So Y-shaped, okay, Y-shaped, and maybe T here. This you can call this a J-shaped branch, and they're non-septate. We don't see any septa dividing the filaments into many cells. So this is perhaps a multi-nucleate cells, 1 cell with many nuclei in it. Okay, there are additional types. These are more complex branching styles. Sometimes you have 2 of the incipient branches that are sort of approaching each other. And I think that might represent an intermediate stage of this A-shaped branch, when the 2 incipient branch eventually touch with each other and then fuse with each other. So this is evidence for cell fusion. Together with H-shaped, for example, here, H-shaped branch and the ladder-shaped branch. These are all evidence for cell fusion, which, by the way, is a fungal feature. This here, again, a ladder type branch. We're going to take a closer look at this. Let me see how to plate this. I need to get rid of my pointer. Okay, here it is. So this is an animation showing that branching structure, the ladder shape. This is done in Andy Zaja's lab using his CLSM confocal laser scanning microscopy. Okay. Next. Additional evidence for cell fusion is this network. So the filaments sometimes form a mycelium-like network. So this also requires the filaments to fuse with each other, and the mycelium is another fungal feature. Not uniquely so. There's some bacteria that can form mycelium-like structure. But I want to also point out some of the larger spheres. So this is a bit larger than the smaller spheres that I talked about earlier. This is about 50 micrometers in diameter. So the two types of spheres, smaller ones, about 20 micron in diameter. They are typically coaxially aligned with the filaments, and we think this could be spore. Reproductive structure of the fungi. The larger spheres is enigmatic. We don't know what to make them. Sometimes you see the filaments penetrate through the larger spheres. It's sort of like a chain of beads type of structure. Two possibilities. One is that there's some sort of symbiotic relationship, two different organisms living in a symbiotic relationship, or perhaps there's some sort of a saprophytic relationship. We know that fungi are heterotrophic organisms, so maybe they're feeding, or maybe they're parasitic on some other organism. Maybe they're feeding on the dead spherical organisms. We don't know. I think this is an animation showing the relationship between the filaments, which is pink, and the large spheres, which is green here. So you can see that some of them penetrate, some of them wrap around, some of them penetrate the large spheres. Okay. The preservation of these filaments, they are mostly pyritized. So this is the two types of filaments, a big and small, or thick and thin. And this is the point where we collect one of the Raman spectrum. This is the point where the other Raman spectrums came from. B here, that's a reflective light image of the same structure in A. The opaque structure are mostly pyrite, some of them reflective. So that's pyrite. That's confirmed by the Raman spectrum, the 344 and the 380 peaks from the pyrite. But there's also carbonaceous material, as evidenced by the D1 and the G1 peaks here. And it's from these peaks we can calculate the temperature, the maximal temperature of this carbonaceous material. So like I mentioned, a couple of hundred degrees Celsius. So our preferred interpretation is that given the several fungal-like features, the thin filaments, non-septate, and possible reproductive structure, and the cell fusion, mycelium-like structure, we tentatively put them here in the within the paraphyletic zygomycete group. So here's your mushroom, here's your yeast, your truffles, and morels. But all these four groups here that belong to the fully terrestrial fungal groups. So the lostated flagellum, that reproduce by spores, as opposed to the aquatic fungi, which has a flagellum and the reproductive cells can move in aquatic environment. And the molecular clock estimate indicate that the origin of terrestrial fungi it depends on which paper you look at, either Tonian or Cryogenian. Okay, so what does this mean? I think the first thing we learned from this, if our interpretation is correct, then there's cryptical terrestrial microorganisms living in these cryptic cavities. Okay. And if they're fungi, that means there must have been other organisms because fungi are heterotrophs. There must be autotrophs to make the organic carbon to support the fungi metabolically. That would mean a more complex terrestrial ecosystem. So it's not just one organism, but a group of organisms that interacted with each other. And I think that point, that interpretation, a complex terrestrial ecosystem, is consistent with the speleothem that we described from the cavities. And the reasoning comes from this. If you look at how modern speleothems are formed, the key process in speleothem precipitation is CO2 degassing. So in modern environment or in Phanerozoic environment, you have this terrestrial biota, soil microbes, and the microbial respiration produce CO2 in the soil, and groundwater penetrate the soil and it picks up CO2, and that leads to acidification and the dissolution of the carbonate. And when the CO2 laden groundwater emerges in the cavity, the gases happen because there's more CO2 dissolved in the seawater than the air in the cave. So degassing of CO2 leads to oversaturation with respect to calcium carbonate. That leads to precipitation of speleothem. So what happened in the soil is critical for speleothem precipitation. This is not the only way to drive non-marine carbonate precipitation, but it is perhaps the most important way to drive speleothem precipitation in modern and in the Phanerozoic world. And we think perhaps this is perhaps indirect evidence for the presence of a soil microbial ecosystem in the aftermath of the snowball Earth. All right. Conclusions and speculations, I must say. So I think we have some direct and indirect evidence for a terrestrial ecosystem in the aftermath of the Marinoan snowball Earth about 635 million years ago. So that indicate that either terrestrial microbes were not affected by the snowball or recovered very rapidly after the snowball, right? And given the resilience of microbes, I think we should consider the possibility that they don't care the snowball. Maybe many of them could survive snowball. So that's another possibility, or they recovered rapidly after the snowball. And we think that shows you the models. I think we should consider terrestrial microbes when we model the post-snowball CO2 drawdown because they are important in break down rocks and consume CO2 after the snowball, particularly when you have a lot of CO2 in the atmosphere. And finally, this is just one data point. And we definitely need more data points to overcome the terrestrial bias. as the Chinese proverb says, wine flower doesn't makes a spring, and I think there's an American equivalent, wine swallow doesn't make a summer. So we need many thousands of flowers, thousands of swallows to understand the terrestrial biosphere in the Ediacaran, particularly during critical time after the snowball. Thank you very much.
Thank you so much, Shuhai. That was very well delivered. So now we're going to open up for our question session. And I didn't detail the question session before like I usually do, but you can ask your question in a couple different ways. You can, if you don't have a microphone or for one reason or another would like to ask your question or would like me to ask your question for you, you can type it into the chat and I will ask it. Otherwise, you can try to virtually raise your hand, which should be an option in the reactions, or you can type a star into the chat if you're struggling to figure that out. And then I will direct you to unmute yourself.
Yeah, I saw Francis McDonald already has a comment in the chat box. He noted that deborites in the back spring are not terrestrial. Yeah, so this illustrates, emphasize the controversial nature of many of the Precambrian terrestrial fossil record. I use this as example. I didn't take a position here. I've not worked on the back spring, so I don't know whether they are or aren't paleokarst. And the same thing goes to Greg Retalic's paleosaur fossils. So I have not looked at them, so I don't know. I just used two examples to show that many of them are controversial.
So Shuhai, I loved it. That was fantastic.
Thank you. Thanks.
Amazing. Thanks, you've obviously considered that these are possibly lichens because the historical connection between the hyphae and the large cells is so, it's a dead ringer for a lichen. Why did you rule out that possibility?
No, I didn't. I said symbiotic relationships is one of the possibility. I didn't use the word lichen, but lichens are basically symbiotic consortium. But I also think it is possible that it could be saprophytic. Yeah, lichen is still a possibility.
I think that's pretty unlikely given the fact that they're enveloping them and they're puncturing them at multiple points. We never see this in a saprophytic fungus. That is really characteristic of the lichen grip.
Okay.
And
well, it's great to know. Good to know.
You should, well, obviously it's a big cell, so it's probably eukaryotic. It'd be good to know more about it, what it actually is. But you might also consider that some of your other even bigger circles might be vesicles, fungal vesicles, very common in the glomeromycota. And these are basically like potatoes. They're dormant phases. There's a lot going on here. I think it's a pretty exciting discovery, and I really congratulate you for it. And particularly Miss Gan, right? She discovered it all.
She discovered this. She's in the audience, so Tian is her first name. Yeah.
Where in Oregon is she?
She's not in Oregon. She's in the audience.
Oh, in the audience. Oh, very good. Yeah. Well, congratulations to her from me. I'll sign off. Thanks.
Thanks.
All right. So Paul Hoffman had told me in the chat that he would like to ask a question, so he looks ready to go.
Yeah, a question for Shuhai and a comment for Greg. First, a question. Why not have the fungi metabolizing marine organic matter that's in the Doushantuo? Why does the electron donor have to be terrestrial as well as the fungi? And the comment for Greg is that molecular clocks indicate that all of the fungal and the algal components of modern lichens originated 300 million years ago. It's not ancient.
Okay, so I let Greg talk about the second, but I can also comment on this. The first one, marine organic carbon, I think it's a possibility. When we submit the paper, one of the reviewers pointed out that zygomycetes particularly needs to have abundant organic carbon, and he or she wasn't convinced that zygomycete, at least modern zygomycete, can survive in low organic content. And given the very low TOC content of the capped dolostone, we think there must be some mechanism to transport organic carbon into these caves, into these cavities. So if this happened before the black shale was deposited in Member 2, then I think the amount of organic carbon in the capped dolostone was not sufficient to support an ecosystem. The second question, and I
A lot of stromatolites.
in the capped dolostone.
Yeah.
Yeah, there's some stromatolites in the capped dolostone as well. I guess it's possible. And the second
water
question.
was hot, so the respiration is pretty efficient. I'll grant you that.
Right. Okay, the second question, let me show you one of the papers that molecular clock of fungi, and so this is a paper published in 2018 that, how do you do this? Share screen.
We don't see your screen.
Okay, here it is. You don't need to know the detail, but what this shows is that the coupling, so on the top, that's a phylogeny and a time-calibrated phylogeny of plants and a green algae. On the bottom, that's a phylogenetic tree and molecular clock of fungi. And this author argued that terrestrialization of plants and fungi happened about the same time. And I think the conclusion is that they are coupled, and it happens in the cryogenian. Big error bar, of course, hundreds of millions of years of error bar. So I don't know. Maybe Greg can comment on Paul's comment that everything evolved about 300 million years ago.
That's strictly lichens.
Yes, it depends on how you define lichen. If you regard lichens as only symbiosis between ascomycetes or basidiomycetes and algae or cyanobacteria, yes, they're younger than 300 million years, for sure, no problem about that. But there are actinobacterial lichens that have been found by the Russians. Not much has been written about them. I can't find too much on them. But there's also this wonderful thing called Geocyphon, which is a glomeromycotin symbiotic fungus, which actually it has within a vesicle Nosticalian cyanobacteria. They're found in forests in Germany. My best guess for that mysterious fossil at 2.2 is it's one of those. Now that has no modern relatives, but it may have had relatives in the Paleozoic. I have several manuscripts out there circulating that suggest that Prototaxides and quite a number of Paleozoic creatures were glomeromycotin lichens with algal or trabuxioid photosymbionts based on their carbon isotopic composition and their morphology. So if you take the strict view that only the modern lichens count and there's no extinct or dinosaur lichens, well, you're right. But I don't think that's a really correct view of the whole picture. And discoveries like Shuhai's are really breaking this whole thing open nicely.
No, I would agree with that. Molecular evidence, of course, is blind to extinct clades. And I was very specifically said modern lichens.
Yes, no,
Yeah.
you're correct. They only go back about 300 million years or so, modern ones.
So it's like
Francis?
Francis McDonald has a question, or Shuhai, did you want to respond to that?
Oh, in the chat box, or
Oh, no. I just thought you were about to start speaking, but.
no? Francis, go ahead.
Hey Shuhai, really nice talk. I really enjoyed it.
Thank you.
I just brought out the point about the debrites in the Beck Spring to sort of back up what you're saying, is it can be really tough to really distinguish between what's terrestrial and what's not terrestrial in some of these localities. And I think along that line, you showed a lot of evidence for the fill within the sheet cracks, the void fill, and that was nice. But you really just showed one sort of picture of what you were interpreting as a karst. And that was kind of central to your interpretation, is that there is some sort of subaerial exposure surface on the top. And I was wondering if you could elaborate a little bit on the evidence that you have for that being a karstic erosional surface, rather than an irregular surface with onlap, because we know a lot of these cap dolostones have very irregular top surfaces, some of which can be submarine from microbial processes, from actual expansion within the sediment. So do you have any evidence for downcutting, karstic fill, grikes, these kind of things that could really give us some confidence that these are actually a karstic surface on the top?
We haven't discovered any grikes, but I think some of the petrographic observations, for example, on the surface of the cavity, you have residual of dissolution. That's microscopic evidence for dissolution. The outcrops in South China, it's mostly covered by vegetation, so it's very difficult to see 3D on the outcrop. Usually you just walk along the road cart. So we haven't seen any grikes or caves that goes into the. But you can see truncation of beds at the erosional surface, or what is inferred as the erosional surface.
I guess what worries me is also the evidence for hydrothermal activity at the time. And I just, maybe if you could elaborate a little bit about how you're kind of reconciling the cement evidence for hydrothermal activity and this sort of paragenetic sequence you're discussing.
Right, the hydrothermal activity in our interpretation happens later. We don't know how late. It could be very late, but lines of evidence indicate that it might be very early, and that's in one of the outcrops. You see that the silica deposit is eroded. It comes to the surface of the cap dolostone, like in a rain, and this could be a hot spring rain. And the silica deposit is eroded and truncated by the overlying sediments. So if this interpretation, that's just one example in a mining pit, so it's difficult to have a more comprehensive investigation of this structure. If our interpretation is correct, then this hydrothermal replacement must also be very early.
All right, great. At the moment, I don't have.
I also have a little modern stalactite to show you the soda straw, so in the middle. So this one is from the Dengyin Formation, a modern one, but in a cave in the Dengyin Formation. It's about maybe 8 or 9 millimeter in diameter. So yeah, and the big one here. With a little hole in the middle, and a little hole in the middle. So yeah, soda straw.
Shuhai,
yeah, Andre.
A good talk. So how deep do you see this dissolution going from the unconformity? And do you see any collapse breccias?
Yeah, they are collapsed structures, and some of the breccia, they are rounded. So not only physical collapse, but also chemical dissolution. I think I have a picture in my talk. I can share it with you very quickly, if I can find it. So they are collapse. Share. Let me go to the end of my talk here. Yeah, here. So you can see this is a piece of dolostone. From the cap dolostone, you can see lamination, and these are all secondary precipitate cements. And you can see the first generation of silica, of what used to be fibrous calcite. So this is more than just physical collapse, but also dissolution, rounding of the corners. A lot of these cracks, they have very sharp corners. So that's probably more or less physical with very little chemical process. But things like this, I think you require some dissolution, some collapse. You mentioned something else.
How deep does?
How deep? Yeah. So the capped dolostone is only about 3 meters on average, and the relief, topographic relief, can be up to 1.5 meters in magnitude. The cavities, for some reason, there's a greater abundance of cavities in the lower half of the capped dolostone. We don't know whether that has to do with the groundwater, the water table that controls on the formation of the cavity or not, and the underlying diamictite has greater porosity that might also have a role to play.
But you had the dissolution and collapse couldn't be hydrothermal related?
Could it be hydrothermal? It has to be
Acidic waters.
No, it has to be earlier because it's coated by ID, and ID is not hydrothermal. Okay. It must predate ID. Yeah, Daryl.
Yeah, my question is the picture you showed of the karst surface is similar to what we called Rundkaran, which is a rounded karstic form that's formed essentially in intertidal settings. And all of the pictures you've showed are consistent with that. There's examples in the Silurian Shikop formation which look identical to that. In that case, you're really dealing with a seawater bathed surface at high tide and low tide. So you may not really have an initial, you may not need a terrestrial organic source for your fungi to work on. It could be an intertidal source.
Okay. Yeah, that's a good point. I think one way to test this would be to look at the geochemistry, like the rare earth element of the precipitate, to see whether it has the marine signature versus terrestrial signature, freshwater, meteoric water.
It's going to be a meltwater signature.
Well, yeah, but compared to you can normalize that against the dolomite, the capped dolostone, right?
Yeah, but the ocean is dominated by the shallow ocean is dominated by meltwater at this time.
So it's an asymmetrical test. If you see that the geochemistry of the cements is similar to the capped dolostone, like the ID, then we would conclude that it comes from this same fluids. But if it's different, then we have a different story to tell, whether it's freshwater or hydrothermal, like Francis alluded to, or marine, intertidal. That's something that need more data, more evidence to be further tested. I think a rare earth element would be a very good tool.
Fair enough.
Do you see any evidence for incorporation of pieces of capped carbonate into overlying sediments, like by subaerial erosion?
So overlying is a black shale in many places, and in Wungan, where we find the fossils, is mostly phosphorite. I must say we haven't paid much attention to the overlying beds. There are no fossils there, and no one has studied them very closely in terms of petrography. But I haven't seen any on the outcrop scale. I haven't seen anything.
We don't have any immediate questions in the chat right now. But if anybody would like to ask a question, you can go. Yeah, go ahead, Paul.
Always asking.
Yeah.
Just a general comment. Glacial rebound, of course, merely brings back the land and sea levels back to where they were before the glaciation. And so to get an emergence, you need an excess of sediment accumulation over erosion during the glacial period, and also minimal accumulated tectonic subsidence. So in this area of the Yangtze platform, the background tectonic subsidence is quite low. And I don't know about Wongan, but Huajibo, the Nantuo is very thick there. And if I recall correctly, the Datangpo is pretty complete. So it could be that there is an excess of sediment accumulation during the Marinoan glaciation over erosion, allowing for the possibility of brief subaerial emergence due to glacial rebound.
Yeah, possible.
That would be an unusual situation specific to this region.
Yeah, there's a paper by J.C. Cravelin a few years back where they modeled sea level change in the aftermath of snowball. My impression is that there's a great deal of spatial heterogeneity. So depending where you are, sometimes you see sea level rise, sea level drop, and it can be up to a few hundred meters.
Be careful, because she did not consider any accumulated tectonic subsidence. She was just looking at the relative sea level change after the glaciation was over. So you have to consider the accumulated tectonic subsidence during the glacial period. That, of course, was not included in her analysis.
Right, but even without that, there's a lot of spatial heterogeneity, and if you're considering what you're describing, the sedimentation, it will only increase the spatial heterogeneity. So it's not surprising that you don't see this at a global scale, only in South China, maybe a few other places. There's some other reports, but Paul, I know you have some different opinions about the interpretation of sub-area exposure in Northwest Canada.
I think Francis McDonald can vouch for my interpretation there.
Can you elaborate that a little bit?
Well, the karst depressions that were reported at the top of the Ravensthroat Formation in the James et al. Paper, if you look at those in detail, those pinched synclines, all the stratification, including the barite layer, is perfectly conformable in both the Ravensthroat and the overlying hayhook. So in fact, those are cuspate lobate folds that are due to tectonic compression of an interface between stiff dolomite and soft limestone. So those features are strictly of tectonic origin.
Okay.
And totally post-date the stratigraphy, which is perfectly conformable.
Okay.
And to add on that, there are other places with these sheet crack cements where there's not evidence of a karstic surface on top, but a transgressive conformable sequence. And so this just gets to sort of the fundamental interpretation of what these void cements are. Do they need to have a subaerial origin or not?
Yeah, in some places it's perhaps purely physical. In South China, I think there's evidence for dissolution. As I showed you, a few lines of evidence, rounding and the dissolution residuals and things like that. I think it'd be very nice to test this additionally using geochemistry to see whether there's different geochemistry from in the cement than the capped dolostone.
On the paleokarst question, Shuhai, if I could jump in the Knuckalina in Australia, there's paleosols in the top of the Knuckalina. It was exposed. I didn't see any caves there, but I did see teepee structures and I did see little cracks. I'm tempted to go back and look more now. But those paleosols were really quite lovely and included petagenic carbonate that was isotopically just like petagenic carbonate, and tau analysis, boron, other indications of freshwater exposure.
The teepee structures are not polygonal in plan view. They're linear and parallel. So they're not teepee structures.
I agree. They're called teepee structures. I just use that because I thought.
Yeah, but don't call them that, because that
Okay, well,
Boron teepees, they are not.
I think they're shadow dunes. Shadow dunes of tufa mounds, as you probably know, because that's the only way you can get that unique alternating structure at the cap of them.
Okay.
Good discussion, guys. I'm enjoying it.
Yeah. Thank you very much. Thank you all the suggestions and the comments. Obviously there's more work to do, and particularly.
But Greg, it can't be terrestrial because it's not red.
Oh, yeah, it is not red.
Oh, go ahead, Walty.
Wait on, it is red. I've been there. I've been to the Doushantuo paleokarst. They are red in places.
Yeah, only because they are red doesn't mean anything.
That's called rose-colored glasses.
You try to leave it right. That's what it's a PMAG.
Vladia has a question.
Yeah, I ordered to comment, I'm not surprised about terrestrial, but it's not only terrestrial, it's subterranean. And that is an important point. We have all those Neo-Archean lacustrine environments and Paleoproterozoic lacustrine environments. There is a hell of a lot of work done on that. And they all show stromatolites, they show microfossils, and so that's not surprising. Surprising, of course, but that is much younger, that you have hyphen of fungi. But I'm not surprised at all that we have all those terrestrial environments at that time, and maybe one should look more carefully and at all those lacustrine near shore raindrop imprint environments within the Neo-Archean and Paleoproterozoic, and who knows what you will find there.
Yeah, and certainly, the lacustrine is equally controversial. I think some of the classical example would be the non-such and the Torridonian, and I think both of them have been challenged. I have not worked on those, but I saw some of the papers recently challenging the lacustrine interpretations. So yeah, it's not easy to interpret Precambrian. You don't have the trees, for example, in Precambrian. So you're lacking.
There is equally a lot of questioning of the marine environments. So that's not an argument, only because someone questions something. The facies analysis, what Sten Avraming did, or what we did in the Hackput or in the Onhaluk, I don't see much of a marine environment there, judging from the sedimentology and the basin analysis. The geochemistry indeed says other things, but those are models. This is not geology.
Yeah.
Lottie, why stop in the Neo-Archean? We could go back all the way to the Paleo-Archean, as you know, into the Moody's group. And Martin Hohmann and myself published 2 papers on these tiny speleothems we had in these silicified sand blisters from an intertidal setting. Remember?
Yes, of course. I just left it out to you because.
All right. Thank you.
Also, there is a paper by Birger Rasmussen in 2009 on 275 hardy formation with thin sedimentary cavity and life and web. I'll send you later.
Yeah, can you also send it to me? I don't think I'm aware of those papers, Andre.
Sure.
Thanks.
Do you have by chance any evaporates in this cap carbonates? Could it be dissolution of evaporates?
Yeah, there's some rosette structure. I don't know whether I can find the pictures very quickly, but the problem is they're in the cave and we don't. It's difficult to constrain their age. Let me see if I can pull out a couple of pictures very quickly. If not, I can send you some pictures and you can. They're no longer gypsum today, but the structures they are rosette. That reminds me it's on my Google Drive, so I think this is from here. Yeah, you guys can keep talking, and I'm looking for the pictures. Here it is. Let me see. Share screen. What do you think about this? Is this something? This is in the cavity, so this could be very recent. I don't know. I have no idea.
Gypsum.
Could be
Think it's a
barite.
gypsum.
Could be barite.
Could be barite.
Yes.
My bet is barite. Yeah. Hear more
Could
of
be
this?
barite after gypsum.
Yeah, there is barite in the cave deposit. Oh, sorry.
These look to me like Marabolite and Senedite.
Can you explain what they are?
Marabolite and Senedite, which are soda salts you get in soils. I've described some from the cryogenian of Hallett Cove in South Australia. They're needle-like crystals that form in soils.
Okay. We have
Barite,
several ideas.
please.
My bet is probably barite.
Barite's generally more chunky than that, like the famous Oklahoma desert roses, the Permian ones. Barite doesn't form needles like that. Narcolite's another one that forms needles, but hexagonal prisms bigger than that.
Those are blades.
Senedite and Marabolite are very, very common in terrestrial efflorescences and in playa lakes.
Okay.
All right. So it seems
right.
maybe everybody is out of discussion points. But
And you announced, Alex, who will speak next week?
Yeah, we talked about that at the beginning. We have only 30 people left here now, but yes, next week is going to be a PhD student from UC Santa Barbara in Susanna Porter's lab. So definitely join for that next week. But Shuhai, thank you so much again for this. This generated a lot of great discussion, and it was really well delivered. So I'm sure everybody here is very, very much appreciated it.
Thank you all. Thank you for your time and your patience. Hope to see you sometime soon in the future in person.
Thanks, Shuhai.
Yeah.
Bye.
Bye,
you.
Paul.
All right, bye everyone.
Thanks.
Bye, everyone.
Bye.